the life of a cell, okay? So this is like the entire lifespan of a cell. And that would be different for different things. So they say that we replace every cell in our body every 7 years, like our whole body is reproduced in 7 years. And I'm like, well, why does it bring the stretch marks back? That's like really unfair. But it's not exactly true because neurons can last our whole lives. That's not true either. So, like the recording won't pick up, but that neurons can't reproduce, and that's not true. They reproduce very slowly. So usually, typically, they will reproduce at a pace that is not going to heal if you get in a tragic accident or exploded or something like that. So the other problem with that, so there was a really interesting lecture down at WSU that was Johns Hopkins University Hospital. He was working on mice, and he would, carefully remove their skull cap and then ping them with a mallet and causing traumatic brain injury, like instantly traumatic brain injury. And then he would watch the neurons heal or reproduce because he's killing off those tissues, damaging the capillaries that feed them and everything. So they have to reproduce to survive. That tissue is dead. What he finds is that neurons do reproduce, but they have scar tissue that impede them from functioning. So when we break our spinal column or break our brains, in not a theoretical way, but like actually break it, then we have these scars that impede normal neurological function. So you can't have your neurotransmitters go and stimulate the next neuron and firing. So he's trying to figure out how to reproduce neurons without the scar tissue. And yeah, but still, nonetheless, like our neurons most likely are still going to be there in 7 years and in 30 years and 50 years. And how you can keep them healthy is, avoiding high fructose corn syrup and other things like that. Exercise, have an active mind, keep everything going. So yeah, whole foods and exercise. It's still the same prescription for just about everything we do. It's unfortunate that chocolate's not really involved in most of life. It's healthy, but like they do not expect you to get your 2,000 calories from chocolate, which is, I feel like it's a sadness, but maybe other people don't. Okay, so this is the whole lifespan of a cell, and it would be distinctly different from cell to cell. So, like, this is not the lifespan of a neuron. This is a lifespan of a typical, how about skin cell? Something like that. Something that's going to have to grow and reproduce. So there's going to be like an entire life where mostly what you're doing is just living. You're just living your life. So I was like, it's not the interstate. What do we call that? I don't even remember. So my brain is just cloudy. It's sad. But we have interphase. That's what it is. It will come. Interstate wasn't that far off. Interphase. So most of your cell life is going to go through interphase, or it's just living its life doing nothing. This little piece of the pie is mitosis. So I drew my original arrow this way, so I'm just like kind of backwards from the way I usually draw it. But mitosis is the active division phase where we form chromosomes and stuff. So we'll break that down soon. But as we are living our lives as cells, we will grow. So you get to a certain point where you're going to take a chunk out and you are going to do G1 growth. So it's just going to grow more. Then we're going to synthesize. So synthesizing is synthesizing both your proteins and enzymes and things inside the cell. You're doubling that, but you're also doubling your DNA. So one copy of DNA becomes 2 copies of DNA. And again, like this is just an interphase where the cell is just living its best life. Then we grow again. So you get bigger because you're literally going to cut yourself in half here soon. So you duplicate all your enzymes, you duplicate your DNA, and just, again, living out your best life. And it's not an exact time model. Like I couldn't put days and hours to these, but there will be some end to growth too, and then it's just going to eventually decide to divide. So mitosis is a very, very, very short part. I would say 5% of cells inside your body will be dividing at any given point. But that is completely off the cuff, not real. But your skin would divide more often, so be in mitosis more often than like a liver cell, something like that. Okay, so we want to take that little piece of the pie, and I divided it into 4, so 4 messy little quadrants. But now we're just going to like kind of put it in a vertical format. So the only things I require you to memorize are PMAT, prophase, so prophase, metaphase, anaphase, and telophase. So we already did our DNA synthesis. That has already happened, where it's double the number of chromosomes or number of genetic content that you had has doubled. But in our normal cell life, so if this was a cell, we would just see our nucleus inside, and it would pretty much be filled in. It would just look darker than the rest of the cell. And what that is that it's our DNA all strung out. It's like just this huge stringy mess, usually, inside here. And that is chromatin. No, I can chromatin. That's right. Okay, so our chromosomes don't form until now. Okay, so we had the giant yarn balls, and it's double the yarn ball there in the chromatin form. But when we start condensing down, so that's taking your DNA and spinning it around proteins called histones, and then those spin around and coil again so that you end up with chromosomal structure kind of like that. So you'll just be drawn as like generic little chromosomes. Okay, so when chromosomes are present, you're in prophase. The other thing about it is that the nucleus kind of just breaks apart. So you might see it. So we'll see like a bunch of chromosomes just sort of like discoordinately like moved about in an almost non-existent nucleus, if there's any one at all. So they're not organized. But metaphase, what you'll see is like a plane, and it's not really a plane, but they're all lined up on it somehow, and chromosomes are just inanimate DNA, right? So we have chemical signals that make this plane exist, and that is metaphase. There's spindle fibers that attach them, but the kinetochores, like the protein centers, are attached to like protein kinetochore fibers and stuff. And there's also chemical signals. And the spindle fibers are actually like attached out here, but the kinetochore is what pulls them to the center. Okay. We ready for the next one? So these are lined up, and why would you want to really closely regulate or orient your chromosomes? So we doubled our DNA, right? Like, so we have 46 chromosomes, and we doubled them all, so we're talking, what is that, 92? 46? I think it's 90, right? So we got 92 chromosomes worth. How easy is that to divide equally into the right locations? I mean, I want to say it's easy, but I can't even like pack a suitcase very well. So I don't think that would be easy with 92 items to perfectly divide. And it's not just perfectly divide. I mean, it is perfectly divide. It's just like it can't just be 46, 46. It has to be the right 46 with the right 46. So you are lined up here very scientifically so that you can start and you have exactly one copy going to this side and one copy going to this side. And like hedgehogs have 95 chromosomes or something like that to begin with. So if we're complaining as humans, it gets worse with other species. Okay, so metaphase lined up along this metaphyseal plate. That's how you recognize that phase. And anaphase is just when they are being pulled. So like they're going in the direction. So you can see the chromosomes not lined up anymore. But they're not like at a pole either. So they're being pulled. So I'm drawing them a little bit differently because they're being pulled in this direction. So they kind of tether or feather in the other way. But they're being pulled to a pole, but they're not at a pole. Like, make sure that we're lining these up right. Okay. Telophase, I think the cheat, honestly, is to see a reforming of the nucleus. Like you'll see like some kind of membranes forming sometimes. But you'll also see like real concentration of the chromosomes at the pole. So they're as far apart as they could have gotten. And I have changed the number of chromosomes so many times that is not accurate, so don't criticize the picture that way. Oh, sorry, we're a bit off screen. Okay, so reforming the nuclear envelope, and you're completely at the poles, so far away, so like a telescope, far away at the poles. For anaphase, I just imagine a little girl Anna just pulling apart the chromosomes. For metaphase, I don't know why it works, to just know that that's the metaphyseal plate, and that works for me. And prophase is, I don't know how I memorized that either, it's just the beginning. So, does anybody have any analogies or things that they use to memorize that? Ooh, metaphase M, so lines up in the middle. Oh, okay. So metaphase looks kind of like a basketball, but I don't know, it feels risky because, I mean, it almost looks like the stitches would be different than on a basketball. But they do touch on a basketball, don't they? Yeah, I'm like visualizing the basketball. Yeah. So basketball metaphor, or that they line up in the middle, and that starts with M2. So how, anybody else have any analogies or things, metaphors? And how does that, like, I mean, that is true. So pulling apart. So I just visualize Anna, like little kid Anna pulling them apart. And here, like telephone, telescope, you know, you see things far away. So they're the furthest point. That's how I remember that. Okay. And then, just to keep them in order, I just memorized PMAT. P-M-A-T. PMAT. Okay, so we good there? You feel confident? Yeah? Yeah? Okay, so we'll stop again so that I remember.